The overall goal of this procedure is to isolate and culture highly purified hippocampal neurons from prenatal mice. This is accomplished by first isolating and dissecting the hippocampus from embryonic mouse tissue. The second step of the procedure is to dissociate individual cells by mechanical force, followed by trypsinization and careful trituration of the brain tissue. Finally, the dissociated cells are counted and cultured using neurobasal media and B27 supplement. Ultimately, purified hippocampal neurons in culture can be used with various molecular techniques, such as immunofluorescence.
Visual demonstration of this method is critical in that it's difficult to visualize the trituration process. The tendency is to be too vigorous when separating the individual cells from the brain tissue. Successful, gentle cell trituration is required to complete the neuronal culture procedure.
To generate prenatal pups for neuron harvest, schedule breeding between adult mice 19 days prior to the day of neuron isolation. Successful mating can be confirmed by detection of a vaginal plug in the female, palpitation, or visual confirmation of pregnancy. The day prior to neuron isolation, for immunofluorescence applications, coat glass cover slips in a 24-well plate with a light coating of 3:1 rat tail collagen 1 poly-D-lysine solution. Spread the solution evenly over the cover slip. Rest the coated plates uncovered in a tissue culture hood under a UV light overnight. The next day, wash the plates with sterile HBSS or store the plates for up to a week at 4 degrees Celsius in the dark, soaking in HBSS. Critical to this operation, sterilize all the instruments and use only sterile gauze. With careful attention to technique, the dissection and harvest of neural tissue can be completed outside of a laminar flow hood. Begin the tissue harvest by decapitating the pregnant mouse at approximately 19 days post-fertilization. Using dissecting scissors and forceps, create an opening in the midventral side of the mouse to completely reveal the mouse's body cavity. Prenatal pups will be located toward the posterior of the mouse's body cavity and should be easily visible in the uterus. With forceps, open the uterus and remove pups. Decapitate the pups with scissors and place the head on gauze under a dissecting microscope. To open the cranium, carefully slice through the skin and cranium using a sharp sterile scalpel or dissecting scissors. Carefully remove the entire brain with forceps and place the brain on gauze. Using a scalpel, remove the cerebellum and incise down the midline of the brain to separate it into two hemispheres. Grasp a small section of meninges surrounding the hippocampus with forceps and pull it gently away to make dissection of the hippocampus easier. The hippocampus is a curved structure that starts in the distal part of the hemisphere and bends ventrally. The inner concave side is facing a ventricle, is the caudal side, and is already free. To isolate the hippocampus, gently lift each hippocampi with tissue forceps and transfer them to a 100 mm tissue culture dish with 3 ml of warmed HBSS under a cell culture hood. Brain tissue can be combined from multiple pups. Using all the sterile precautions possible, under a cell culture rated laminar flow hood, use a scalpel to gently mince the brain tissue in HBSS. Transfer the minced tissue and HBSS to a 15 ml conical tube. Add 1.5 ml of HBSS and 0.5 ml of 0.25% trypsin solution to a total volume of 5 ml. Cap and gently invert the tube 4 or 5 times. Avoid producing bubbles or the tissue may become trapped on the bubble and not settle to the bottom of the tube. Incubate the tissue at 37 degrees Celsius for 15 minutes, inverting tube as before every 5 minutes. After the incubation, carefully pipette away the solution, leaving the tissue at the bottom undisturbed. Now, Wash the tissue with 5 milliliters of HBSS 3 times. Allow tissue to completely settle to the bottom of the tube between each wash, and carefully remove the wash solution. After the final wash, resuspend the tissue in 2 milliliters of HBSS. Before beginning trituration steps, prepare fire-polished Pasteur pipettes. Hold a sterile 9-inch Pasteur pipette tip in a flame until diameter of the pipette opening is approximately 0.5 millimeters and the edges are slightly rounded. Too large an opening will make trituration inefficient, and too small an opening will damage the cells, so be careful. Using a normal sterile 9-inch Pasteur pipette, gently triturate the tissue a total of 7 times. If this process is performed too vigorously, the cells can be damaged and die. Allow larger tissue pieces to settle to the bottom of the tube prior to moving to the next step. Transfer the supernatant to a fresh sterile 50 milliliter conical tube. To the remaining tissue, add 2 milliliters of sterile HBSS and triturate a total of 5 times using a fire-polished Pasteur pipette. Allow all remaining larger tissue pieces to settle to the bottom of the tube and combine supernatant with the previous supernatant for a total of 4 milliliters dissociated neuronal cells. Count the dissociated cells using a hemocytometer. As a general rule, once cell numbers have been determined, subtract 20% from that final number to account for any cell death that may occur after plating. Mix the cells with neurobasal plating media. For 24-well plates, prepare to add 60,000 cells per well in 0.5 milliliters of media. For 60 millimeter culture plates, prepare to add 400,000 cells in 3 milliliters of media. For 100 millimeter culture plates, prepare to add 6 million cells in 6 milliliters of media. Swirl plates gently to distribute cells evenly. Place neurons in a 37 degrees Celsius, 5% carbon dioxide incubator overnight. The next day, remove half the volume of media from the cells and replace it with the same volume of neurobasal feeding media. Unlike plating media, this media lacks glutamate and high donor horse serum. Neurons should be fed every 4 days by removing half of the old media and replacing it with the same volume of fresh neurobasal feeding media. Neuronal processes should begin to be visible on day 1 and become extensive and branched by day 10. Sometimes, contamination by glial cells can occur. They are easily distinguished from neurons in culture, and in this instance, they make analysis of a GFP-LC3 expressing neuronal process difficult to visualize. To reduce glial cell contamination, treat the culture with cytosine arabinoside before the 4th day of culture at its lowest effective dose. If the culture is contaminated by fibroblastic reactive microglial cells, treat it with FRDR to decrease their proportion. Hippocampal neurons were grown for 7 days and stained with a vital dye to mark active mitochondria and tissue culture cells. The cells were fixed in 4% paraformaldehyde PBS and visualized by fluorescent microscopy. The dye itself is non-fluorescent until oxidized in the mitochondria. Active mitochondria can be seen throughout the neuronal processes. Cells from the 7-day culture were also fixed with 4% paraformaldehyde PBS and immunostained with monoclonal antitubulin antibody, followed by Oregon green labeled goat anti-mouse secondary antibody, and fluorescence visualized by microscopy. Hippocampal neuronal cultures were grown for 5 days and transfected with GFP-LC3 beta DNA construct using lipofectamine 2000. At day 7, the cells were fixed using 4% paraformaldehyde PBS, and agrosomes with GFP-tagged LC3 beta incorporated into their outer membrane were visualized using fluorescent microscopy. Agrosomes are located throughout the cell body and neurites and are denoted with arrows.
While attempting to perform this protocol, it's important to remember to perform the procedure quickly but carefully and to be gentle with the cells during the trituration process. Paying attention to these variables will greatly enhance your ability to successfully culture mouse hippocampal neurons.